despair, okay. Hopeless, sure. Some fatigue, drained, panicked, angry, okay. Surprised, cheerful, thrilled, a little cluster here at pleasant, restful, complacent, I think, sleepy. So seeing the spread, but definitely feeling today the vibe is very low energy, maybe more on the unpleasant side. So check in yourself, see if that's where you need to be today, see if there's anything you can do to kind of get you into a mood that's going to be conducive to this class or to the rest of your day. Is there anything that's going on that I should know about? Midterms. So grades were posted yesterday. If you do have any questions, the answer key was also posted as well, so please refer to that answer key. And if you do have any questions, please come and see or book a time during office hours or come see Dr. Patterson and I during office hours as well. If your grade is not posted, there's two reasons. One, you wrote during the makeup. That means that we're still processing those and those grades are going to appear a little bit later. Or there's an issue with your Scantron and you need to come and see us so that we can verify your Scantron and double check that with you. So those should be the only two reasons that you don't have a grade unless you didn't write completely. All right, so myself, where am I at? Right now I'm going to say I'm more in the high energy pleasant. I'm going to go with just, I don't know, let's see. I'm going to go not too high, maybe pleasant. I'm just going to go with pleasant. That's where I am. All right, today we're going to continue our discussion around carbohydrates and the metabolism of carbohydrates. We're really going to focus today on glycolysis, focusing on how the cells extract energy from those carbohydrates in the first part of these metabolic processes. So at the end of this lecture, I want you to focus on being able to summarize how glycolysis is able to break down glucose and extract energy from it. Key word there is summarize. You're going to see I don't need you to memorize all of the glycolytic pathway, but I do want you to understand what's happening in that pathway. I want you to be able to examine the role of thermodynamics as a driving force for glycolysis. So how do we go from glucose to pyruvate, and why is that energetically favorable, allowing us to release energy? I want you to be able to describe how glycolysis can be regulated depending on the metabolites in the pathway, their concentrations, how this can shift the pathway in different directions, but also is going to start to regulate some of those regulatory enzymes. And how cellular ATP concentrations, or the overall energy state of the cell, is also going to have an impact on whether glycolysis is turned on or turned off. And then finally, we're going to look at hormonal regulation. We started to see how insulins can regulate the transport of glucose into the cell. Now we're going to continue to look at how insulin and glucagon and epinephrine, these hormone regulators, are going to turn on and turn off enzymes within the pathway to adjust the flux, or how quickly glucose is going to be processed and converted into pyruvate. So this is a slide that I had later in the lecture. I brought it up here, duplicated it here as well. We're going to come back to it again. I just want to emphasize one key point. Metabolism is an overarching word to describe all of the anabolic and catabolic processes that are occurring in the cell. So metabolism is kind of everything. We can have anabolic processes that make things, take small building blocks and make things that are bigger. That's what we call an anabolic process. And we have catabolic processes that take big things and break it out into smaller things, often to release energy. Now, both these processes will be occurring in the cell at any given time. When we're building something up, we need energy to do that. So we're both going to have anabolic and catabolic processes turned on. And we're going to see how glycolysis is a catabolic process, but it's turned on during that anabolic state. When we're talking about an anabolic state within the cell, this is when the cell has lots of resources, has lots of energy, has lots of building blocks. So it's going to take those building blocks, take those resources, and make things with it. That can be protein synthesis, this can be cell division, making nucleotides, gene expression, storing some of that extra resources as glycogen or fats. This is how the cell is going to carry out those anabolic processes, but this requires some energy. The main hormone you're going to see during this anabolic state that we're going to talk about, at least mostly, is going to be insulin. Insulin is very important for regulating how glucose gets used within the cell and within the body. We saw insulin's regulation of glucose transport on Tuesday. We're also going to see how insulin regulates the enzymes within the glycolytic pathway. And it's going to be a common theme throughout, seeing where insulin pops in to signal to the cell to use the glucose, to use energy to make things. So an anabolic hormone, or signaling an anabolic state. But glycolysis is still going to be an overall catabolic state, because it's going to take glucose, a 6-carbon molecule, and break it down into 2 smaller molecules of pyruvate. When we're in a catabolic state, so there's low amounts of glucose, low amounts of energy, this is going to signal to the cell to start breaking down some of those molecules that have been built up, maybe energy that's been stored as glycogen or stored as fat, or maybe proteins that are going to be not going to be made, and protein synthesis might decrease, and you might see a net effect of protein breakdown, all in a way to maintain homeostasis in the cell. So in that catabolic state, different hormones turn on to signal the state, epinephrine and glucagon. Looking at how glucose metabolism fits into this process, we're going to see 4 different pathways we're going to talk about. We're going to look at glycogen synthesis, so building up glycogen and storing that glucose. Glycogenolysis or glycogen breakdown, breaking down that glycogen to release glucose back into the cell. So we're going to see an anabolic and catabolic form production and breakdown of glycogen. Glycolysis, which is going to be the focus of today's lecture, where we're going to break apart glucose and release energy. And then on Tuesday, we're going to talk about anaerobic metabolism very briefly, just what happens to pyruvate when we don't have oxygen around. So today we're going to focus on glycolysis. The question I always get is, what do you need to memorize and what do you need to know? So I'm going to be very explicit. You need to know the structure of glucose and pyruvate. The other metabolites in between, you don't need to memorize their structures. You need to know the total substrates and the products of the pathway. So what is the total input and what is the total output? And we're going to highlight these in later slides. You need to know where and why the pathway branches. So looking at that aldolase step, where you're going to break apart glucose, or what was that glucose molecule, into those 2 3-carbon molecules. And you need to know which enzymes are regulated and how they're regulated. So this is what I want you to memorize and focus on for memorization. But the overall understanding of the pathway is where you're truly going to be assessed. So an example, connecting back to, if I wanted to connect back to our first learning objective, so to summarize how glycolysis breaks down glucose for energy production. Here's a Mentimeter question that addresses that learning objective. Which one of the following statements is correct regarding glucose? Just get the Menti going. Is it that glucose produces H2O and CO2, so water and carbon dioxide, as the products of glucose breakdown? Glycolysis is an anabolic pathway that requires ATP. The glycolytic pathway includes 10 enzymatic reactions that produce 1 molecule of pyruvate. Or glucose and ATP availability both regulate glycolysis. So which of these statements is correct? Survey says majority going with D, glucose and ATP both regulate glycolysis, and that would be correct. So looking at this question, what's wrong with option A? Glycolysis produces water and carbon dioxide as a product of glucose breakdown. We're going to see in a few slides, but what you may remember from previous courses and previous learning is that the product of glycolysis breakdown is 2 pyruvate molecules. The water and CO2 are going to come through in later products where the pyruvate is now broken down and oxidized fully to carbon dioxide in the TCA cycle. And water is going to be generated throughout the pathway, but one of the key areas where water is going to be generated is at the end of the electron transport chain. So while glycolysis does lead to pyruvate and eventually to those molecules, water and carbon dioxide are not products of glycolysis. B, glycolysis is an anabolic pathway that requires ATP. An anabolic pathway is something that builds something up, and glycolysis is a catabolic pathway. It's going to break glucose apart. So it does require ATP, but it's a catabolic pathway. Option C, the glycolytic pathway includes 10 enzymatic reactions. That part's correct, but it produces 2 molecules of pyruvate, not 1, so that's going to be an incorrect response. And as we'll see as we go through these later slides, the availability of glucose, how much ATP is in the cell, is going to regulate whether glycolysis is turned on or turned off. So let's take a look at glycolysis. In summary, glycolysis is going to be a 10-step catabolic pathway found primarily in the cytoplasm. This is a key point of where these glycolytic enzymes are located within the cell. Because they're in the cytoplasm, when glucose gets into the cell through those transporters, all the enzymes are there available to start to break down glucose. Another important piece is that it doesn't require any other organelles in order to process glucose. It doesn't require mitochondria, doesn't require any other organelles or systems to break it down. All of this is present in the cytoplasm. So cells that don't have a mitochondria can still use glucose for energy. This is a key point when we think about red blood cells. They don't have any organelles, they don't have a nucleus. All the enzymes that are going to be required to break down glucose are present within the cytoplasm of those red blood cells. This is also why we see the glycolysis present in almost every organism, even lower bacteria. The start of that process starts with glucose. Glucose that gets into the cell, remember glucose is a hexose, has a 6-carbon molecule, a sugar molecule. This is going to be the starting point for glycolysis. The first stage is all about preparing that glucose molecule to be broken apart and separated. This is where energy is inputted into glucose. It's going to be phosphorylated twice, generating a bisphosphate or a 2-phosphate molecule. And that fructose 1,6 bisphosphate gets separated into 2 3-carbon molecules. We're not losing anything here. We're not losing anything as carbon dioxide. Those carbons are still maintained. That separation separates it into two eventually identical 3-carbon molecules, glyceraldehyde 3-phosphate, that both follow the same pathway through the second stage of glycolysis. And this is where energy is going to be extracted. The energy extracted here is through what we call substrate level phosphorylation. The difference here, it means what we mean by substrate level phosphorylation is that the phosphate that's added onto ADP to make ATP is coming from a substrate. It's being transferred from one substrate to ADP to make ATP. This is in contrast to oxidative phosphorylation, which occurs in the electron transport chain. There, ATP synthase takes a free phosphate plus ADP and smushes them together to make ATP. So this is two different levels of phosphorylation, or two different types of phosphorylation reactions to generate ATP. At the end of this process, you're left with two 3-carbon molecules of pyruvate. These pyruvate molecules can have different fates at the end of glycolysis. In cells that don't have a mitochondria, or when there's not a lot of oxygen, they can be metabolized to lactate through a process called anaerobic metabolism that we're going to talk a little bit about next week. When there is a mitochondria available, and when there is oxygen available, that pyruvate is going to enter into the mitochondria and be oxidized through the TCA cycle and into eventually through the electron transport chain to generate carbon dioxide, generate ATP, generate water. So depending on the cellular conditions is going to dictate the fate of that pyruvate. But focusing on glycolysis, that glycolytic pathway, in summary, starts with charging or priming that glucose molecule to be broken apart, and then extracting energy from those molecules to generate ATP. Overall, coming back to thermodynamics, this pathway is going to be exergonic. Exergonic meaning it releases energy. And coming to the first law of thermodynamics, energy can't be created or destroyed, it can only change form. That energy is captured in ATP. So we're releasing energy through the breakdown of glucose and capturing that energy or storing that energy as ATP and NADH that can be used later on in other parts of the pathway or used by other enzymes or other molecules in the cell. Looking at the 10 steps, again, you don't need to memorize all the 10 steps. Understand where it's going to split and focusing on the structure of glucose and pyruvate, so the beginning and the end. The overall process is going to generate use 2 ATP molecules in the first half, generate 2 NADHs in the second half, and generate 4 ATP in the second half as well. And each one of these steps are linked together in the cytoplasm. The product of one reaction becomes a substrate of the next reaction, coupling these enzymatic reactions together. But on Tuesday, we were talking about other types of hexoses that we're going to get from our diet. We don't just get glucose, we also get galactose and fructose. Those molecules have to enter into the same glycolytic pathway. Galactose, again, is going to be an isomer of glucose, needs to be converted into glucose, or glucose 6-phosphate, and then it gets metabolized through the same glycolytic pathway. It enters into the pathway a little bit later, but it eventually follows the exact same process. Likewise, fructose is either going to be converted into fructose 6-phosphate, entering into the pathway, or it's going to be broken down directly into DHP and glyceraldehyde 3-phosphate as well. This allows our cells to be able to use glucose, galactose, or fructose for energy and extract the same amount of energy regardless of the initial form of that carbohydrate. Remember, these are all isomers of each other, all 6-carbon hexoses. Fructose is going to be a ketose, galactose and glucose are going to be aldoses, but they're all going to come into the same pathway and merge, and they're going to go through the same second stage of that reaction, extracting the same amount of energy. Looking at the overall reaction, here's a summary of these 10 steps of the glycolytic pathway. Note for information only, I don't need you to remember every single reaction in the pathway or the standard energy conditions. What I want you to know about is the total sum of the substrates and products of the reaction. So if you add all these reactions together, you'll see that one glucose molecule is going to react eventually, or in sum, with 2 NAD plus molecules, 2 ADP molecules, 2 free phosphates to produce 2 pyruvates, 2 NADHs, 2 ATP, 2 hydrogens, or 2 protons, and 2 water molecules. None of these atoms are lost, they're just changing forms. That 6-carbon glucose is converting to those 2 3-carbon pyruvates. Looking at the Gibbs free energy in standard conditions, so under laboratory conditions, recall that standard conditions here indicate 1 molar concentrations, allowing it to come to equilibrium, 25 degrees Celsius, pH of 7, 1 atmosphere of pressure. Under those conditions, some of these reactions are going to be energetically favorable, so they're going to move in the forward direction. Some of them might be a little bit closer to reversible. However, this is not what's happening within the cell. In the cell, concentrations or all these reactions are now linked or coupled together in a pathway. The substrate or the product of one reaction becomes the substrate of the next. This adjusts those concentrations of those molecules to make this more favorable in a single direction. So while the overall free energy change under standard conditions would be about 35, you can release about 35 kilojoules of energy. Under cellular conditions, this is going to release a total of about 76 kilojoules of energy because of the linking and coupling of these enzymes. And if you're looking at the free energy changes, 3 processes should stand out to you. The first step, hexokinase, is going to be energetically favorable. It's going to release a lot of energy. Step 3, a phosphofructokinase, is going to release a lot of energy. And the last step, pyruvate kinase, is going to release a lot of energy. Looking at this energy release, because they're so exergonic, they're going to be essentially irreversible. They're only going to move in the forward direction. Every other step within this pathway, when we put them into a cell, has a free energy change that's relatively or close to 0. This means that it can be reversible and will depend on the concentrations of the relative concentrations of the substrate and product of which direction they go. This is important to note because these pathways that release the most energy, that are essentially irreversible, are the pathways or the enzymatic reactions that become regulated by the cell. Something that's irreversible is the thing that you need to stop in order to stop the pathway. So looking at how this coupling looks like maybe graphically in another way, under standard conditions, we can see the energy changes over time when these reactions are seen in isolation. Starting from 0, the free energy change between reactions will go up or go down. And gaining energy is really difficult and requires usually an energy input to allow that molecule to get to a higher energy state. This is unfavorable and can be really challenging when you're looking at these reactions in isolation. Kind of like trying to ski uphill. It's very difficult. However, when we start to put them in a cellular context where these reactions now are coupled together, we can see the effect of this pathway, coupling it, is that it's essentially a net downhill, skiing downhill or like a waterfall. These reactions flow together when the product of one becomes a substrate of the next, pulling these molecules through this enzymatic pathway to their eventual conclusion as pyruvate. Those big changes in energy and those key steps are where we capture, or where we're able to capture, essentially capture much of the energy required to generate those ATP molecules later on. So looking at these 3 steps, the first step being hexokinase or glucokinase, depending on the cell that you're in. Step 3, phosphofructokinase 1 and pyruvate kinase. These are where you're going to see the regulation of glycolysis. Because they are essentially irreversible, these are where the cells have evolved to stop it. The first step requires ATP, or an input of ATP, and the second step as well requires an input of ATP. Thinking potentially from an evolutionary point of view an advantage, why would the cell waste energy generating or breaking down glucose when it doesn't need it? These require ATP, so if it's trying to conserve ATP, those steps that require ATP are the ones that are going to be shut down. The last step, pyruvate. If it doesn't need pyruvate, doesn't need energy for that pyruvate to enter into those later cycles, let's turn that process off and turn the whole pathway off. Looking at how these pathways are regulated, you're going to see some common themes. 1, the products of the reaction are able to regulate the enzymatic reaction. These are often through 2 different mechanisms. 1, the product can bind to the active site and block the substrate from being able to bind. Or 2, the product can bind to an allosteric site, changing the conformation of the enzyme. By changing the conformation of the enzyme, it's going to inactivate the enzyme. So as the product builds up and is not being used in later reactions, it'll turn the enzyme off. A little bit of a feedback regulation. You also see other allosteric regulators from different parts of the cycle or from the cell that's going to indicate either high energy states or low energy states. These allosteric regulators can now turn on the pathway or turn on the enzymes or turn them off depending on cellular needs. The third regulation we're going to see is post-translational modifications and often phosphorylation. So this is a modification or a phosphorylation that happens to the enzyme, allows it to undergo a conformational change that puts it into its active form. Often these phosphorylation reactions are going to be regulated by those hormones, insulin, glucagon, and epinephrine, to turn on enzymes or turn them off. And the other key piece is glucose. If there's no glucose in the cell, like from those glucose transporters are turned off, or the concentration of glucose is so low that it's not moving into the cell, this pathway doesn't have a substrate to begin with, and this pathway is going to be turned off. So glucose availability is also going to be a key regulator of glycolysis. So let's look at the steps of the pathway. Step 1, hexokinase. In some cells, you also see this referred to as glucokinase, or not referred to, a different enzyme called glucokinase. Hexokinase is useful because it works on multiple hexoses in order to phosphorylate it. So potentially fructose, galactose, glucose. It does have activity for all of these depending on based on the shape of its active site. Glucokinase only works on glucose. It doesn't work on galactose, doesn't work on fructose, and we saw a reason why looking at the active site on Tuesday when we looked at the active site of glucokinase. This is a key step because it traps glucose in the cell. Those glucose transporters that we talked about, they work by moving glucose along its concentration gradient. If the concentration of glucose in the cell increases or gets to a high point, glucose can actually move back out of the cell along its concentration gradient back through that transporter. However, this first step in the glycolytic pathway, hexokinase, will phosphorylate glucose, converting it to glucose 6-phosphate. There's no transporter for glucose 6-phosphate, so this traps it within the cell. Because this reaction is also essentially irreversible, that glucose phosphate now needs to be used by the cell in some process. We will see an example later on of a different enzyme in the liver cells that can dephosphorylate this, a separate enzymatic reaction that allows the liver to be the one tissue that can now release glucose back into the bloodstream to regulate blood glucose levels. But we're going to talk more about that one on Tuesday. So now that glucose is trapped within the cell, that glucose 6-phosphate needs to be used in one of a number of pathways. The main one we're talking about today is glycolysis. So now it's going to be broken down to release energy. But it can also be used potentially for glycogen synthesis. If glycolysis for energy is not needed by the cell, those later steps in the pathway are turned off. Glucose 6-phosphate will build up, and now it's going to be stored as glycogen. And again, we're going to talk more about that on Tuesday. It can also potentially be used to make other metabolites for different processes, in particular, the pentose phosphate pathway. The pentose phosphate pathway, pentose, thinking of 5-carbon sugar molecule, this helps to generate the riboses and deoxyriboses we need for DNA. So this allows this glucose molecule to be used in many different pathways for many different metabolites. But once it's in the cell, it's going to stay in that cell and be used there. Looking at hexokinase, this molecule, this enzyme, like glucokinase, they're very similar enzymes, work almost like a hinge. The binding site for glucose is in the center, but there's also a binding site for ATP on what we call that large domain, away from the binding active site. When this molecule or glucose binds, that conformational change bends the two parts of the protein together and brings ATP in close proximity to glucose, snap, allowing that reaction to occur and transferring that phosphate. So this conformational change is essential for allowing this reaction to occur. Looking at the structure, so this is just a different representation of the enzyme active site. We can see the binding sites for, let me change this, for ATP and for glucose. So when they bend and fold over, bringing those into proximity. That glucose 6-phosphate, now the product of the reaction, can stay stuck in the active site or bind to the active site if the concentration increases. And this starts to turn off hexokinase. When we turn off this enzyme, glucose is no longer phosphorylated, and glucose can now exit the cell to go back into the bloodstream. So if the cell doesn't need any more glucose, it allows it to return to the blood to be used by other tissues. Glucose 6-phosphate can also bind to an allosteric site away from the active site, causing that conformational change, causing hexokinase to fold in a different way that's not going to be as active. And then insulin here is the first place where we're seeing our hormone, our anabolic hormone regulating this pathway. But this doesn't do it directly. This does it by regulating its gene expression. So when insulin is expressed in the cell, it binds to that insulin receptor on the surface, initiating that signaling cascade inside the cell. That you're going to see that regulating some enzymes directly through phosphorylation. But in this case, it actually turns on the expression of hexokinase. Coming back to our enzyme lecture, remember that hexokinase or gene expression. When we're thinking about how the Vmax of a protein or of an enzyme, the more enzyme we have, the higher the Vmax. So by initiating gene expression, you are increasing the amount of glucose that can be phosphorylated within the cell, trapping it in there. So insulin works in that anabolic state by increasing the total amount of enzyme present. Looking at the mechanism, this is just for information only. When they fold to bring it into close proximity, we see a nucleophilic attack on that gamma phosphate. So we label these as alpha, beta, and gamma. The last phosphate is a gamma phosphate. So this glucose molecule will attack it, one of the hydroxyl groups, removing that phosphate group to generate glucose 6-phosphate. So a phosphate at the 6th position. Remember 1, 2, 3, 4, 5, 6. So a phosphate at the 6th position of glucose and generating that ADP. The next regulatory step is the third step of this pathway, phosphofructokinase. This also requires ATP. So this is where the cell is going to now continue to activate this hexose in order to produce, eventually prepare for breakdown. This works through a similar mechanism where you're going to see one of the hydroxyl groups attack that gamma phosphate on ATP and transferring it over to now the number 1 position on fructose. PFK1 can be regulated in a lot of different ways. One, it can be inhibited allosterically, so ATP, and we'll see citrate, can potentially bind to the allosteric site, turning it off. When there's lots of ATP, this is indicating the cell has lots of energy. Doesn't need to be activating glycolysis, and it can turn off this pathway. Citrate, we'll see, is a metabolite of the TCA cycle. Again, when those concentrations are getting high, it indicates there's either a backlog or there's a lot of energy available. We don't need any more ATP to be generated. We can turn this pathway off. It can also be activated allosterically by either AMP and ADP. So again, these are going to be the metabolites of ATP that have lost phosphate groups, so indicating the cells may be in a lower energy state. This is going to activate the pathway, and it activates it by binding to the allosteric site of phosphofructokinase and turning it on. Another allosteric activator is fructose 2,6-bisphosphate. And this is an important activator as well, but it's a little bit harder to think about why this might be important. So fructose 2,6-bisphosphate comes from a side reaction. As the concentration of fructose 6-phosphate increases in the cell, that indicates that phosphofructokinase maybe isn't working fast enough. Other enzymes can start to use this metabolite as well. In this case, phosphofructokinase 2 is going to phosphorylate fructose 6-phosphate on a different site, on the number 2 position. This is using up some of that substrate that's present as the concentration increases. Now, this new metabolite that appears within the cell actually has a very important function. It's an allosteric activator of phosphofructokinase 1. By turning on or generating this metabolite as the concentration increases, it starts to increase the speed or increase the rate of PFK1's activity, allowing it to metabolize that fructose 6-phosphate faster, generating this next substrate. So when the concentration builds up, we get this side reaction. This is where we start to see our hormonal regulation come into play again. Insulin is going to activate this enzyme, and you're going to see glucagon turning that enzyme off. Thinking about what's happening during this metabolic state, insulin's indicating lots of glucose is available. It wants to increase flux or flow through this metabolic pathway. So it's going to turn on this reaction in order to activate PFK1. Glucagon is going to signal a low energy state or low blood glucose levels. It wants to decrease this pathway, so it turns off this metabolic pathway. Insulin binds at an insulin receptor, initiates that signaling cascade. Glucagon is going to bind to GPCRs and activate a separate signaling cascade to turn it off. We're going to see this again a little bit later on when we talk about gluconeogenesis with Dr. Patterson in a few weeks. But it's important to understand how these pathways are all kind of merging together. Now, that allosteric regulation has some important considerations. Because ATP is both a substrate and an inhibitor, the level of ATP, or the concentration of ATP, the relative concentrations of ATP and fructose 6-phosphate are going to impact the activity of this enzyme. Looking at this progress curve of this reaction, we see the reaction velocity, or VI, and the concentration of our substrate over time. When we have a lot of ATP available and low substrate concentration, this enzyme activity is going to be relatively low. It's going to turn off. That ATP is binding both potentially to the active site, but also to that allosteric site. As the concentration of fructose 6-phosphate increases, now we have lots of fructose 6-phosphate in the active site, lots of ATP in the active site. It's going to be more difficult for that ATP to bind to the allosteric site and inhibit the enzyme. So we're going to start to see this enzyme get to its maximal activity. Likewise, or conversely, for ADP or AMP, which are allosteric activators, when we have lots of those around, they are not binding to the catalytic site. They're only binding to the allosteric site, activating this enzyme. So at all levels of substrate concentration, this is going to ramp up the activity of the enzyme and turn it on by binding to that allosteric site. So I have a question for you then. Thinking about fructose 1,6-bisphosphate, so this is now the product of PFK1. Data has shown, looking at how this impacts the cellular homeostasis, that when this molecule increases in concentration, it activates RAS. Now, RAS is an important protein because it's going to stimulate cell growth. Why is this significant, but not necessarily surprising? Why is it not surprising that fructose 1,6-bisphosphate might stimulate growth in the cell? So looking at the response coming in, energy is needed for growth, indicates a level of ATP, lots of energy available. Absolutely. It's active, there's lots of energy available in the cell when the cell can grow. Absolutely. So thinking about what this indicates in the pathway. When this metabolite starts to increase, it indicates one, there's lots of glucose available, it's being converted to glucose 6-phosphate, and it's coming through the pathway. This is now starting to activate all these other proteins, getting them ready to start using this energy and these metabolites and starting to use glucose to help the cell often to grow. So it's a signal to the cell that this is a high energy state, this is an anabolic state, let's go. Let's start using all these resources. Yep, just the article. So step 10, the last step that's being regulated in glycolysis is pyruvate kinase. So this is the final step that's going to transfer that last phosphate group to ADP to generate ATP. This is going to result in the product pyruvate, which is the final product of the glycolytic pathway. Now this is going to be a highly regulated step within the pathway. We can see regulation at multiple levels. One, through those hormones, insulin and glucagon, but also through a number of metabolites that are going to turn this pathway on and off. This is important because at the very end of the pathway, if we can stop production at pyruvate kinase, the concentration of every single metabolite starts to build up. Remember, those are reversible reactions until you get back up to PFK1 and then eventually hexokinase and be able to turn it off. So this is important in just kind of being the final stop and just shutting down the whole pathway. Two ways that can be regulated are through phosphorylation. So this enzyme is directly phosphorylated to turn it on or turn it off. Whenever you see high blood glucose levels, you should immediately be thinking insulin. When insulin binds to the receptors, that insulin receptor on the surface of the cell, activating that signaling cascade, it's going to activate a protein phosphatase. That phosphatase can remove the phosphate from pyruvate kinase, turning it on. This is a key step in now activating that pathway. When you're thinking low blood glucose levels, you should be thinking glucagon. When it binds to its GPCR, so it's glucagon receptor on the surface of the cell, tied to that G protein, it's going to activate a protein kinase A. And that's going to lead to the phosphorylation of this and an activation of this enzyme. So these hormonal regulations can turn on and turn off this enzyme when needed, when indicated by the total glucose supply within the body. We can also see the metabolites regulating this protein. Fructose 1,6-bisphosphate, so we just saw how that's going to regulate RAS and indicate total cellular growth, that there's resources available to grow. This is also going to activate this process. It's going to stimulate the conversion of pyruvate, or phosphoenolpyruvate to pyruvate to generate that metabolite, getting the cell ready to use all these resources, use energy, use these metabolites available. In terms of inhibitors, unsurprisingly, ATP. If you have lots of ATP, you don't need to be generating more energy. The cellular levels of ATP is actually kept relatively low. So when ATP levels get so high or too high, all these pathways start to turn off, so we're not generating ATP that's not needed by the cell. This is binding to an allosteric site. Pyruvates, also a potential allosteric inhibitor. So this is a product of the reaction. It can bind potentially in the active site, turning it off or blocking the ability of phosphoenolpyruvate to bind. But it's also combined on another site to keep it in its inactive state or to change its conformation. Alanine as well. We're going to talk about alanine in a second, but alanine can also inhibit this pathway. And then you also see AMP and acetyl-CoA. Now some reports are going to see AMP and acetyl-CoA as allosteric regulators. Some of it is more indirect regulators. But either way, the levels of AMP and acetyl-CoA are also going to regulate the activity of this protein. But coming back to alanine, why do you think alanine is going to regulate pyruvate kinase? We've seen other metabolites, ATP signaling high amounts of energy. We've seen metabolites in the glycolytic process or pathway turning on and turning off the metabolites. Acetyl-CoA even makes sense because it's going to be the next step in the pathway. But why alanine? What's alanine doing here? So seeing a lot of responses coming in. DNA replication, not so much. Small molecule that binds, acts as a ligand, yes, technically. Production of ATP, because of the structure, similar structure to pyruvate. Now this is a key point. Remember earlier in the course, we asked you to know the structures of your amino acids because there's going to be a lot of things that can be synthesized from them. This is one of the key things when we're thinking about alanine and pyruvate. Alanine and pyruvate are essentially very similar in structure. The only difference is the presence of that amino group. And this allows pyruvate and alanine to be interconverted into one another. If you have lots of pyruvate, it's a reversible reaction. If you have lots of pyruvate, it'll be converted into alanine. Likewise, if you have lots of alanine, it'll be converted into pyruvate. So any of you who are taking those protein shakes with lots of those amino acids, lots of alanine in there, this is what's happening to some of those proteins as it enters into your glycolytic pathway, and you can burn it for energy. You'll see another amino acid here, glutamate. Glutamate serves as the amino donor here. So it's donating its amino group to pyruvate to generate alanine. And it's generating another molecule called alpha-ketoglutarate. And we're going to see that again in the TCA cycle. So this allows metabolites from the amino acids to now enter into these glycolytic pathways or these energy metabolism pathways to generate energy. So it's important to think about what's happening with allosteric regulation. Allosteric regulation is all about changing the conformation of a protein. It binds to somewhere other than the active site, changing that conformation, and turning that protein off. Here's an example of pyruvate kinase. In its active conformation, pyruvate kinase is a tetramer. There's four monomers come together to form this active tetramer, each with its own active site. However, when these allosteric regulators bind to pyruvate kinase, it changes its conformation and doesn't allow it to form this active tetramer structure, this quaternary structure. It now forms a dimer complex, and that dimer is inactive. So when those allosteric activators leave the allosteric site, it can reform this active structure, this tetramer, and carry out its function. Think of it as the best example I can think of off the top of my head right now is you working at your desk, typing, working on your computer, and someone putting something on your back and pulling your arms back, making it really difficult for you to work, little T-Rex arms. That can be a challenge, and it's changing the conformation of that enzyme, or in this case, your body, making it more difficult for you to be able to do your job and to work. Same thing is happening with these enzymes. So, checking in now on inhibiting glycolysis in this regulatory pathway, what do you think will be the most efficient way to regulate? I'm going to go back here. What do you think is going to be the most effective way to inhibit glycolysis? Do you think it's that feedback inhibition of hexokinase with glucose 6-phosphate? The allosteric regulation or inhibition of PFK1 by ATP levels? Is it that covalent modification of pyruvate kinase? Or inhibiting glucose transport? What's going to be the most effective way to turn glycolysis off? A little bit of a spread, but looking at most of the responses coming in, going with D-ish, which should be correct. If there's no glycolysis, or there's no glucose, there's no glycolysis. So that doorway into the cell can just turn off the whole pathway because it doesn't even have a substrate to start with. Everything else is just fine-tuning the flux in the pathway. This binding interaction between ligands and proteins is kind of like an on-off mechanism. Some of it's going to be bound to the enzyme, turning it off. Some of it's going to be unbound, allowing the enzyme to turn on. But if you want to shut down the pathway completely, you stop glucose. That's why glucose transporter 4 is so effective at stopping muscles and adipose cells from using glucose. There's no way for those cells to even get it to use that pathway, forcing them to use fatty acids. So summarizing how the regulation occurs, in glycolysis, we're converting those monosaccharides to energy. It's a very favorable catabolic pathway. If the cell has enough energy, glycolysis is going to slow down. It doesn't need to generate those energy, so the metabolites or those enzymes are going to be turned off. But however, when the cell doesn't have enough energy, this gets ramped up very quickly. Those metabolites, or anything that indicates a low energy state, glucagon, ADP, AMP, insulin, ADP, AMP, will potentially turn on this pathway. A lot of the regulatory mechanisms through feedback inhibition, allosteric regulation, post-translational modifications, but these all focus on regulating those spontaneous, irreversible reactions. And when it's inhibited, that glucose has to go somewhere. It can either potentially leave the cell, go back into the blood, but in cells that are able to, like the muscle and liver cells, this is going to store that glucose as glycogen. And we're going to talk more about that next week. So just to summarize, metabolism is going to be that sum of those anabolic and catabolic pathways, but those anabolic pathways require energy, so you still have some catabolism happening at all times, including glycolysis. Insulin is going to turn on this pathway, taking that dietary carbohydrates, converting it into glucose, and taking some of it, and we'll see storing as glycogen, epinephrine is going to turn on, or glucagon are going to turn off this pathway, allowing you to break down that glycogen. So some key messages, but we're going to continue to talk about this next week.